everyone. I wanted to start with a question to you. Did you ever wonder how we sense temperature, how we sense that it's hot, how we sense that it's cold? Well, many of you know probably that the senses that we get from the outside world through our eyes, our hearing, our taste, and what we see, what we feel through our skin, reaches our brain through neurons, and that's what relays the signal from outside world into our brain. But how does this occur on a chemical molecular level? How is this external signal translate into chemicals that could actually reach the brain? So at the edge of a neuron, which is a very long cell, you have many membranes, many membrane proteins. Where's the laser? Oh, okay. You have many membrane proteins, and each membrane protein is responsible for different stimuli. So there are membrane proteins that could sense temperature, and there are membrane proteins that could sense mechanical pressure and differences in pH and so on. And how do they work? So these proteins are often ion channels, which means that they have this sort of tunnel-like structure, and it's usually closed. Now, when you get the external stimuli, which in our case is heat or cold, these proteins, they open up, and they allow ions, such as sodium or calcium, to enter the cell. And the change in the concentration of these chemicals inside the cell triggers an action potential, triggers the neurons to fire, and send the signal to our brain. And the discovery of the proteins that are responsible for our sensation of heat or cold was done by David Julius here, and he was awarded half of the Nobel Prize in 2021 for the discovery of receptors for temperature and touch. And so this is a quite hot subject. So there are many different types of trip channels. So there are specific trip channels for every range of temperature. I think it's interesting to note that the feeling of cold is not the absence of a feeling of heat, but rather there's a specific protein for every temperature range, for cold, warm, and hot temperatures. And trip channels are also activated by various natural compounds, such as garlic and chili pepper and mint. So the next time you eat something spicy, you know that you're actually opening up a trip channel. Now, trip channels, are, because of their importance, they're actually well studied, and there are currently more than 200 structures of 20 different trip channels. So you could see different examples of structures here. Now, they're all a bit different, but the common feature for all of them is that they're tetramers. They're all built of 4 subunits. Now, you could ask, why is this important? Why do I want to know how the protein is looked? Why do I need to know that it's made of 4 subunits? Well, if you want to understand how a protein functions, what's the mechanism by which it operates, you need to know its structure. For example, if you want to know how human beings walk, you need to know that we have legs. And then another reason is that often in disease, when you have different diseases, it's caused by mutations in these proteins. And these proteins often affect the structures of the protein. So if we go back to the example of the leg, we wouldn't know that the leg is broken if we didn't know how it looked when you're healthy. So we need to understand protein structure to understand how it works in health and disease. And indeed, trip channels are involved in many different diseases, many skin diseases, such as atopic dermatitis or eczema, but also in cancer and asthma and hypertension. So you really need to understand how trip channels work. Now, like I mentioned, there are a lot of information about trip channels. However, there's this one phenomenon that's a mystery despite more than almost 20 years since its discovery, and that's the pore dilation phenomenon. And this phenomenon is observed when you activate certain trip channels, either for a very long time or much more intensely than you usually do. And in those cases, scientists observe that the trip channels do not only allow passage of ions, these small atoms, but they also allow passage of small molecules. So much bigger entities could now enter the trip channels, and that these ions and small molecules could now flow in both directions. And the only possible explanation for how this happens is that somehow the trip channel expands and gets larger. But when you look at the actual structures of the proteins that have been discovered, and you look at the profile, the diameter of the pore of the trip channels, it's not big enough to allow passage of these small molecules. There's only room here for ions, for little atoms to go through, not for molecules. So how does this happen? So to try and address some of these questions, I used in my postdoc a technique called high-speed atomic force microscopy, or high-speed AFM. This is a technique where you take a very sharp tip, you pass it over a surface, you get a topography of the surface you're imaging, and you could get an image. Now you get these images very, very quickly, so you could get actual videos. And you could get videos of protein in action on a single molecule level. You could see also conformational changes and assembly. So we use high-speed AFM to study the TRPV3 channel. So what you see here are many TRPV3 channels inside the membrane. This is a view from the top. So it's a topography image. And you could see that many of these proteins are tetramers. They're made of 4 subunits, like we expected. However, what was surprising to us was that here and there we could see pentamers. We could see proteins that are made of 5 subunits. Here you could see it in higher resolution. That was very weird. And even a weirder observation was that we could see that these states are reversible. So we could see tetramers that are stable for a few minutes and then suddenly transition into a pentamer. Here you could see here another example. And we could also see pentamers that switch back into tetramers. So this is a pentamer, and then it goes back to 4 subunits. And again, we see the same thing. So then the question is, what is the purpose of this pentameric state? Could this be related to the pore-dilated state? There were 2 reasons that made us think this. One is that from what we saw in the high-speed AFM, these pentamers occur on a rather long time scale. Only after a few minutes you start getting them. And pore dilation occurs over a prolonged stimulation, also on the order of minutes. And 2 is that, like I said, pore dilation, you have to have a larger pore. And when you look at the AFM images, you could see that the pentamer has a larger pore than the tetramer. So to check this, we thought, well, could we perhaps intensify the signal in our AFM images? So we added an activator that when you increase the concentration of this activator, it causes normally pore dilation. So we added to our AFM system, and we counted the number of pentamers versus tetramers, and we could see a 2-fold increase of the number of pentamers when we are in pore dilating conditions. So that's one good news. And the second sort of goal that we had was to try and solve a high-resolution structure of the pentamer and see if indeed we could see that the pore is big enough to accommodate these small molecules. To do that, we used the technique of cryo-EM, where you take an electron microscope and you take many images of your protein in different orientations, and when you average all these orientations, you get a high-resolution structure. So we collected many of these images, about 10,000 of these images. You see here many, many molecules of protein. Now, the first straightforward structure that we solved was the structure of a tetramer. This is still the more predominant state. But when you look carefully, you can see that here and there are some pentamers. Here you can see them on the sides. And after a very lengthy process, I succeeded finally in getting different orientations of these pentamers and in solving the structure of the pentamer. Now, when you compare the structure of the pentamer to the structure of the tetramer, you could see that indeed the pentamer has a much wider profile of the pore, and this is big enough for small molecules to go through. So it seems really that the pentamer is the pore-dilated state. Of course, there are still many more questions like what's the physiological role of this pentamer? We currently think that it might be related in disease. So certain diseases are caused by increased occurrence of pentamers. So there's still a lot more to research, but this is some sort of beginning in trying to understand this phenomenon. So to summarize, TRP channels are responsible for our sensation of temperature. Usually, TRP channels are tetramers, but we discovered the first instance of a pentameric TRP channel. And we show that the pentamer appears to be the pore-dilated state. So with that, I'd like to acknowledge you, acknowledge my group for my postdoc. And thank you for your attention.